Hello, this is my biology role model project, and the biologist I decided to make a video about is Carolyn Bertozzi. Carolyn Bertozzi graduated from Harvard University with a bachelor's in chemistry, and she got her PhD at the University of California, and she did her postdoc in the University of California in San Francisco. Right now, she is a professor at Stanford University. One of her hobbies is playing music, and during her undergraduate, she played in several bands, including Board of Education, which she played in with Tom Morello, who went on to become the lead guitarist for Rage Against the Machine. Something that I admire about her is that she was the first openly LGBT person to win a Nobel Prize. About 43% of queer scientists are closeted, and because of stereotypes, usually when people think of a scientist, they don't think of a queer person. So it's just really special that a queer person had won a Nobel Prize. The specific field of biology she studies is biochemistry. She studies sugars found on cell surfaces and how they impact human health. And she also invented a whole new field of science called bioorthogonal chemistry. This is an area of biochemistry that's concerned with changing the molecular structure of biomolecules in a way that lets you observe biochemical processes better, but doesn't interfere with the processes themselves. And this is demonstrated really well in this experiment of hers. From mechanism to mouse, a tale to bioorthogonal reactions. So this figure shows the results of two different experiments, but for the sake of time, we'll just focus on one. The two bars on the left of the graph show the results of an experiment where a Western blot was performed on two groups of cells, meaning they exposed the cells to a specific antibody that recognized the activity of the enzyme HRP. Now, the interaction between the antibody and the enzyme gave off a signal that could be measured as light. Two groups of Jurkat cells, which is a type of T cell, were grown with or without the type of modified glycoprotein. And the graph below shows the amounts of activity they were able to measure in the control and treatment groups for both experiments. Now, the modified molecules could not influence the biological processes in the cells, and so this means that the activity was a lot easier to track using the modified molecule. In conclusion, Bertozzi has uncovered so far a groundbreaking way to track biochemical interactions, and she is also applying this to medicine and is using her knowledge to find cancer treatments.